Hello, my name is Grace Yu, and today I will be presenting my MiCM Summer Scholars Project. My supervisor was Dr. Nicole Lee Jessen, and the project's name was Computational Modeling for Intervertebral Disc Biomaterial Design. Now imagine staring at a screen for hours and hours in a day. Well, you probably don't have to imagine this right now, but a common complaint is lower back pain. Lower back pain is caused by intervertebral disc degeneration or disease. And intervertebral discs are gel-like tissues that live between tough, bony vertebra and the spine. If an intervertebral disc doesn't do its job because it's damaged or diseased, the vertebral bones rub against each other, causing pain and damage to nerves. A current solution is to have surgery to fuse vertebrae together or hold them in place with metal bars and screws. Unfortunately, this is very invasive and not always successful. That's why our collaborators proposed using biomaterials as treatments instead. Biomaterials are substances that are engineered to mimic tissue. For example, there are gels for intervertebral discs or hard scaffolds for bones. Intervertebral disc biomaterial could be injected between the vertebra to regenerate or replace intervertebral disc. The problem is that there are many recipes for biomaterials. For example, you can base them off of alginate from seaweed, chitosan from fungus, or many different synthetic substances, and adding these in different ratios can make substances ranging from soft gels for vocal folds to hard scaffolds for bones. In addition, people add different types of cells that could be native to the tissue or outside of it, and chemicals like growth factors to stimulate those cells. And all of these factors make designing biomaterials more difficult. That's why I worked on making a computational platform for testing biomaterials virtually. Basically, we want to take a biomaterial recipe and be able to predict its outcomes. A biomaterials recipe includes its composition or basis, like alginate or chitosan, cells included in the biomaterial, and chemical concentrations of any growth factors, pro and anti-inflammatory mediators, or cytokines that you want to include at the start. The outcomes include mechanical variables of the biomaterial, such as stiffness or porosity, cell populations, and chemicals that, for example, could be produced by those cells, like growth factors and other mediators. For all of these, we're interested in seeing how they change over time and also a spatial picture of what's going on. For example, if there's too much stress in one area in the biomaterial, that could be a problem as well. And so we can also split this model into two different aspects. A biological one to see if the body accepts the biomaterial, if cells can live in it, and if they can regenerate intervertebral disc tissue. And also a mechanical aspect to see if the biomaterial can withstand the stresses in your spine. The biological aspect, like the cells and extracellular matrix proteins and other chemicals, are simulated with agent-based modeling, which is a common technique for simulating biological phenomena. And the mechanical aspect, including viscoelasticity and diffusion, needs to be simulated by another type of model that's better suited for physics. And in this case, it's finite element modeling. And beyond this, there are many interactions and much overlap between the biological and mechanical aspects. For example, cells can respond to mechanical stresses, and cells produce substances that can harden or soften biomaterial. And so, with all of this complex modeling going on, we need to improve the model's accuracy by optimizing and refining it with data from the bench in an iterative process. The end goal is to be able to find the optimal biomaterial recipe to treat lower back pain. So here are my references, and I would like to acknowledge especially the MiCM summer scholars who were my colleagues and also who supported us in this. Thank you for listening.